Hey, it's Mr. Peterson. This screencast is about the transition between DNA, which is an information storage molecule, into the business of making proteins. And we're going to need a different kind of nucleic acid. We're going to need RNA, which is ribonucleic acid. So it's kind of the same, but it's a little bit different. Now we'll get into some of the differences. So transcription, so we've got replication. That's DNA making a copy of itself. Transcription is DNA being converted into RNA. So we're going to start with DNA, which is a gene, right? So we've got a sequence of DNA that has some information on it, and we're going to turn it into a single-stranded RNA molecule. So here's that DNA from the bioluminescent jellyfish. We can take that gene and we can put it on all kinds of things. So here's a little roundworm, little ascarid, or we can make fruit flies glow, or we can make fibroblasts, which are cells in your body. We can make them glow, or we can make mice glow. We've talked about that. We can make mushrooms glow. We can make plants glow, even aliens, right? Your favorite movie, Avatar. Probably a little bit dated now. You probably have never even seen Avatar. But anyway, DNA is universal. It's universal, and since we can make copies of it, if we can make copies of it, the DNA is going to code for the same kind of information in all kinds of organisms. So that's bioluminescence. Pretty cool stuff. So here's the central dogma, or this is the big idea of genetics. So, and it's all about information flow, and it's unidirectional. It goes one way. So we have DNA, which is an information storage molecule. Right, that's stored in the nucleus of eukaryotic cells or in the nuclear area of prokaryotic cells like bacteria. It gets converted into a single-stranded molecule called RNA, ribonucleic acid. So it gets converted. The DNA stays there. The information is then transferred to RNA. And that sequence of RNA is actually a sequence of amino acids. And amino acids are the things that make up proteins. So the information flow, here's the central dogma. We go DNA, RNA, proteins. That's really what it's about. So keep that in mind. DNA, RNA, proteins. Turn it into a song or something like that. I probably should do that, write a song about that. Okay, so the central dogma includes three processes. It includes replication, and so DNA makes a copy of itself so that cells can keep making more cells that make the same kind of proteins. All right, that happens in the nucleus. It happens in the S part of the cell cycle, right? So we talked about that in the replication screencast. Transcription happens when we're going from DNA to RNA, when we're going to need to make proteins. So when a cell is done with replication and made a copy of itself, when it's done with cytokinesis, then DNA is gonna say we need some parts for this new house that we just built, and transcription is going to lead the way to making proteins. And that process of going from nucleic acids to amino acids is called translation. We're changing languages. We're getting away from the nucleic acid language into the language of proteins. That's the next screencast in this series, translation. All right, so an RNA is the connection. It's the link between nucleic acids in the storage phase, which is in the nucleus, into the proteins, which is the action phase. It's where the work is being done, since the cells rely on proteins to do work. Okay, so RNA is that big link. All right, so RNA is a little bit different from DNA in three ways. So let's see if you can recall. What are the three building blocks of DNA? What is a nucleotide made of? See if you can recall that. Pause and write that down. Draw a picture of one, maybe. Well, I'll pause. There you paused now. Okay, RNA is the same kind of thing. It has a sugar. It has, instead of the base thymine, it has uracil. So it has a U instead of a T. It's a single ring structure, just like thymine was. So it has to be the same kind of shape, and it's a single-stranded molecule. Now, it can fold back on itself, but it's just a single long chain of nucleic acids. So it has a phosphate that connects to sugars, that connects to ribose sugars, and it has uracil instead of thymine, and it's single-stranded. So there's kind of, it's folded back on itself. You can see it's a single strand if you follow that, but it also folds back on itself to give it some three-dimensional shapes to it. All right, so transcription makes three types of RNA. The first one is mRNA. M stands for messenger. So messenger RNA is going to take the information from the nucleus out into the cytoplasm of the cell to build proteins, where it's going to get translated. Ribosomal RNA, the ribosomes are actually the machines that build the proteins. So rRNA, rRNA is, again, single-stranded. It just folds back on itself to give it a three-dimensional shape, three-dimensional conformation. Those are the machines. Cells are going to have billions of, I'm sorry, that's a total lie. Cells are going to have thousands of ribosomes in them, depending on the kinds of cells. And different cells that make proteins could have more ribosomes than others. So cells that don't build a lot of proteins, maybe you're not going to have as many. And that could be like tens or thousands of ribosomes. And then we've got tRNA. And tRNA is it's transfer RNA. It brings the amino acids to the ribosome to build the chain of amino acids. So we're starting to talk about transcription. Transcription uses RNA to go from DNA into a different kind of conformation. So here's a little animation of transcription. Here's our DNA, and we've got another molecule of polymerase, and this is an RNA polymerase. And notice it's a little bit different. It's a different color. So it has U instead of T. It matches up the same. If you want to stop and rewind that a little bit, you can do that. We've just built a single strand of mRNA. You'll notice that it only copied one half of the DNA. That's what it does. That's called the sense strand. The sense strand has the gene. The other side is called the nonsense, or sometimes called the antisense. So only half of the DNA is actually the gene, and the other half of the gene, scientists believe, is there for protection, to kind of guard the gene. So transcription does convert that single chunk of DNA into a single strand of RNA molecule, and so it doesn't matter if it's tRNA, mRNA, or rRNA. They're all produced the same way. It's similar to replication because there are enzymes and proteins, complementary base pairings. A pairs with U instead of A pairing with T because there's no thymine in RNA. But there's G and C, so that complementary base pairing rules. They have different end results. The DNA makes a copy of DNA, and transcription just copies the gene. It makes a single strand. Replication makes one copy. Transcription, depending on the need of the cell and how much protein they need, they could make multiple copies because they can crank those out and produce lots of protein depending on the cell or the organism's needs. So transcription, the DNA from mRNA, it's a little bit different. Here's a picture of DNA being transcribed, and all of those things that are coming off so that's a single gene, but all of those things coming off are RNA molecules. So these center strands of DNA, multiple copies of RNA, the cell must need a lot of this particular type of protein. So growing RNA strands in transcription. So transcription, a little bit different than replication, but it has some of the similarities. Enzymes and proteins doing some work there. So transcription. Hope that helps.